Hello again. Thank you for coming back for our first official speaker of the day. I am now proud to introduce Dr. Mary Ellen Urick for today's webinar on informing end-to-end cancer drug development with bioinformatics. Dr. Urick earned her doctorate from Cornell University, demonstrating the role of cyclooxygenase enzymes in hen model of human ovarian cancer, and subsequently studied the clinical relevance of pharmaceutical agents as radiosensitizers and the functional relevance of genetic mutations present in endometrial cancer. Today, she will be speaking on the use of transcriptional signatures for potential clinical trial population selection. And now I would like to turn it over to Dr. Urick for her presentation.
Thank you, Sandy. Thank you for that introduction. And I also would like to thank everyone who's taking time today to listen to this presentation. As Sandy said, today I will be describing the potential to use transcriptional signatures for clinical trial population selection. So I wanted to give you a brief outline of what I'm going to discuss today. I will start by giving you a definition of what I mean by predictive biomarkers, and I'm going to do this by defining what a predictive biomarker is compared to other common types of biomarkers. Then my talk really is going to be broken into two main sections. In the first section, I'm going to highlight our expertise in generating transcriptional signatures through a service offering that we like to call OncoPredictor. What OncoPredictor is essentially broken up into three phases. Within the first phase, we call that the cell line response phase. We do an in vitro screening of a cell line panel that can be a number of different sizes. We typically like to use about 240 cell lines. The second phase of the project is known as biomarker discovery. And this is where we would generate these transcriptional signatures that I will describe today and focus on today. But we also identify a number of other univariate as well as multivariate biomarkers. In the final phase of OncoPredictor is where we do clinical mapping. And this is an effort to identify clinical populations that do express the predictive biomarkers that we identified in phase 2 of the OncoPredictor. So the second half of my talk, I'm going to focus on our expertise in generating signatures now using the Oncoming knowledge base. And of course, I'm going to describe the Oncoming knowledge base for you in more detail, but just briefly what it is the compendium of the world's genomic data that we have been collecting and curating for the last 8 years. It contains over 70,000 clinical samples and also contains preclinical samples, including cell line as well as xenografts. Throughout the talk, I'm going to demonstrate the idea that these signatures really have the potential to help build competitive differentiation plans during drug development. Some of the real value that we see in these signatures, particularly in an offering such as OncoPredictor, where you're using an in vitro panel. If you're doing univariate biomarker discovery, you really would be limited to just the biomarkers that you have already identified within the cell line panel. But these signatures have the potential to really represent the true biology of what's going on after drug treatment. And the final bullet point I have here, for the sake of time, I'm probably not going to touch on it today, but I did want to let you know that these transcriptional signatures we find can be useful in identifying potential therapeutic combination opportunities as well. And as I said, I probably won't touch on it today for the sake of time, but I'd be happy to discuss this in follow-up with anyone who's interested. So what I have on this slide is examples of 3 common types of biomarkers: diagnostic biomarkers, pharmacodynamic biomarkers as well as predictive biomarkers. And what I think of when I define these biomarkers is diagnostic biomarkers identify patients that need treatment. So they're kind of the who needs treatment. Pharmacodynamic biomarkers, on the other hand, identify what dose the treatment should be given at. And predictive biomarkers, which is what I'm going to focus on today, identify patients that should respond to a particular treatment. At the bottom of the slide, what I'm giving is examples of a number of different aberrations that could be used as predictive biomarkers. And we here at Thermo Fisher Scientific have expertise in utilizing many, if not all, of these aberrations as predictive biomarkers. But as I described today, I really am going to focus on gene expression alterations and in particular transcriptional signatures and their use or their potential use as predictive biomarkers. And so the general idea is that the use of predictive biomarkers is expected to lead to enhanced response rates in clinical trials. And I'm sure anyone that has worked in drug development knows how critical it is to get the desired response rates within your clinical trials to enable FDA approval of your drug and to get this drug into the hands of physicians and which then can be used clinically. And so the general idea is if we have a hypothetical cohort of cancer patients, they will harbor a number of different genomic alterations. Some of these genomic alterations could confer sensitivity to a targeted therapy, but some of these alterations could actually confer resistance to that same targeted therapy. So the general idea is if you don't stratify before giving a targeted therapy, you could get a perceived low response rate. And that is because your patients that do actually harbor the alteration that confers sensitivity to your targeted therapy are diluted among the general population of patients that either don't harbor that alteration that confers sensitivity or harbor another alteration that actually confers resistance to that targeted therapy. So the idea is if you take that same hypothetical patient population and define biomarkers and stratify based on those biomarkers before giving treatment, you may get the same response rate as you did in the example on the left, but now, for example, in the hypothetical patients shown in dark here, you have a perceived enhanced response rate because these patients are no longer diluted among the general population. There are examples in clinical trials of predictive biomarkers actually being effective. I'm sure many of you know of HER2 positive patients responding to Herceptin, as well as EGFR mutated patients responding to erlotinib, just to name two. There are also a number of clinical trials currently ongoing that are trying to determine whether or not predictive biomarkers and targeted therapies actually lead to enhanced response rates in clinical trials. And at least at the time of ASCO 2014, which I had the opportunity to attend, many of these clinical trials were giving encouraging results, indicating that predictive biomarkers and targeted therapies do actually lead to enhanced response rates in clinical trials. So here at Thermo Fisher Scientific, as I may have mentioned already, we do have expertise in the identification of predictive biomarkers, and we have a number of different service offerings that we can use to help our clients build competitive differentiation plans. But today, I'd like to highlight our OncoPredictor analysis. As I mentioned previously, this analysis is broken up into 3 phases. The first phase is cell line response. The second phase is biomarker discovery. And the final phase is clinical populations analysis. We have had clients use this sort of OncoPredictor analysis very early in their drug development programs when it might be most optimal to think about predictive biomarker identification. We've also had cases where we come in retrospectively, where perhaps someone did not have the best results in phase 1 clinical trials, and we come in as a sort of rescue mission to identify predictive biomarkers for their particular drug. And as I've said, we've had success in both of these scenarios in identifying biomarkers that could potentially help in clinical trial design. So in the first phase of the OncoPredictor analysis, this is where we do our in vitro screening of a cell line panel. We work very closely with a number of collaborators, and here I'm highlighting our close collaboration with Urick and Pan Labs, where they would characterize response to our client's drug in 240 cell lines, for example. They then collect a number of different cell proliferation endpoints. I'm giving the examples here of 5x apoptosis, GI50, EC50, as well as IC50. And then once we receive the cell proliferation data, we then review and QC all of the response data. And then we evaluate a number of different call strategies to determine the optimal categorization of cell lines as either sensitive or resistant to the particular therapy. This stage of the OncoPredictor is very important that we identify the correct categorization of cell lines as sensitive or resistant, because moving forward into phase 2 of the OncoPredictor, only the cell lines that are characterized as sensitive or resistant are used in that phase. And I would like to point out that we work very collaboratively with all of our clients. We make sure that we are aligned with them within each phase of the OncoPredictor analysis. And so this would be an example of a portion of the analysis where we would come to agreement with our client as to whether we have optimally categorized the cell lines as sensitive or resistant. So if you look to the left of the slide that I'm showing right now, at the figure that I'm showing there, that's an example of results that we could get in the first phase of OncoPredictor analysis. So on the y-axis, we would place your endpoint there, be it 5x apoptosis, GI50, etc. And then each of the bars on the graph represents an individual cell line. And we have it colored by response to the drug. So cell lines that we've designated as sensitive are indicated in pink, and cell lines we've designated as resistant are indicated in green. Moving on to phase 2 of the OncoPredictor analysis, this is where we would integrate the genomic data of the cell line panel with the sensitivity and resistance calls that we've made in phase 1 and come to alignment with our client. We evaluate a number of different biomarker types within this phase 2. They include mutations, amplifications, deletions, and a number of different expression-based aberrations, including our custom sensitivity and resistance gene signatures, which I'm going to highlight in the talk today. So on the next slide, I actually am going to describe to you in detail how we derive these signatures. We perform both univariate as well as multivariate biomarker analysis, and we use a number of different analysis tools to prioritize which biomarkers will move forward into phase 3 of the OncoPredictor analysis. So again, if you look to the left of the slide that I'm showing right now, the waterfall plot at the top is the exact same waterfall plot that I showed you on the previous slide, and now below that, I have a waterfall plot that's now indicating either the presence or absence of a particular biomarker. So in pink indicates that the biomarker is present, and blue indicates that the biomarker is absent. And so what you can see is by focusing in on this right-hand side of this graph, I hope you can appreciate that we have a pretty sensitive biomarker of resistance that has pretty good specificity. And what I mean is the majority of the cell lines that express this biomarker are resistant cell lines, if you look on the right-hand side, whereas we're not picking up very many of the sensitive cell lines colored in pink with this biomarker. As I said, within phase 2 of the OncoPredictor analysis, this is where we derive our custom transcriptional signatures. We essentially do this by completing a differential expression analysis of the sensitive and resistant cell lines. We then use p-value that is derived according to a student's 2-class t-test to rank the genes. The top 1% of genes that are differentially upregulated in our sensitive cell lines then becomes our sensitivity signature. And the top 1% of genes that's differentially upregulated in our resistant cell lines becomes our resistant signature. At this point within the OncoPredictor analysis, if we did identify the transcriptional signatures as significant predictive biomarkers, we would put the signatures through a sort of robustness testing to make sure that the signatures are valid and we feel confident in moving forward to stage 3 of the analysis with these signatures. So for example, one of the easier robustness tests we use is leave one out cross-validation. Should the signatures pass our robustness testing, we could then move forward to stage 3 of the OncoPredictor analysis, which is clinical populations analysis. And what I'm going to illustrate on this slide is one example of one of the analyses we could do within stage 3 of the OncoPredictor analysis. And this is what we like to call our UBER analysis. UBER stands for U133 Based Expression Resource. And what it essentially means is we are just using a portion of the Oncoming knowledge base of samples that have been analyzed on U133 chips. So essentially what we can do within this analysis is we can bring in our sensitivity and resistance signatures, and we do this by z-score normalizing the expression values for each of the genes present within the signature. And then the mean z-score expression values across all of the genes in the signatures becomes the sensitivity and resistance scores for the clinical samples that are found within the UBER database. The UBER database contains both clinical as well as preclinical samples, but for all of the examples that I'm going to show you within this slide deck, I have filtered our UBER database to just show clinical samples. So if you look at the figure on the right, what I have on the y-axis is an example of if I imported a sensitivity signature expression. And on the x-axis, I have placed our resistance signature expression. Each of the points on the graph represents an individual clinical sample that is colored by cancer type. And then the dark lines on the graph represent the pan-cancer median expression of the sensitivity signature and the resistance signature. So what I hope you can appreciate from this is that we can very quickly, just by using this analysis, focus in on, for example, the upper left-hand portion or the upper left quadrant of this figure. And let me see if I can highlight this. This quadrant is what we would describe as clinical samples having favorable signature expression. And what I mean by that is that they have high sensitivity expression and low resistance signature expression. And so again, just making the point that through this analysis and through the use of these signatures, we have the potential to very quickly help our clients focus in on clinical populations, be it by cancer type or cancer subtype, that could potentially respond to their drug. And so, as I said, the UBER analysis is one example of an analysis that we can complete during stage 3 of the OncoPredictor process, which is clinical populations analysis. But really in this stage, we are able to use any of the internal tools and power tools we have to use to integrate the entire Oncoming knowledge base. So we use all of these different resources to map the biomarkers that we identified in stage 2 of the process to cancer types and cancer subtypes. During this phase of the analysis, we can also perform hypothesis-based biomarker investigation, should it be requested. And we can also identify additional cell lines for biomarker hypothesis testing, again using the Oncoming knowledge base since it contains both clinical as well as preclinical samples. And so I wanted to give you an example of some actual results that we could obtain using the OncoPredictor analysis. Of course, the majority of the results that we have derived using this analysis are proprietary client information, but we have completed a number of OncoPredictor projects as proof of concept. And so I'm going to share with you just a little of those results today using the MEK inhibitor CI1040 as an example. So for the sake of time, I'm kind of jumping forward a little bit in this analysis. So I've skipped the cell line response phase, and I'm jumping right into biomarker discovery. So it's phase 2 of the OncoPredictor. In the figure that I'm showing you here, on the y-axis, I have indicated p-value. And on the x-axis, I have indicated odds ratio, with a positive odds ratio being associated with sensitivity biomarkers. So these biomarkers that you see kind of shooting off to the right of the graph would be our sensitivity biomarkers. In contrast, a negative odds ratio is associated with our resistance biomarkers. So the biomarkers you see shooting off to the left of the graph would be our resistance biomarkers. And I have p-value decreasing as you go up the graph, so meaning the further out the biomarkers are and the higher up they are, the more significant the biomarkers would be. So each of the points on the graph does represent an individual biomarker that is colored by the type of biomarker it is, which is indicated in the legend. And it's sized by the number of times it occurs within the cell line panel. So what you can see, what I hope you can appreciate here, is that we did identify as some of the most significant biomarkers, predictive biomarkers that you might expect would confer sensitivity to MEK inhibition. This includes RAS-RAF pathway mutations as well as MAP kinase pathway mutations. What I'm indicating here with the arrows is that the most significant resistance biomarker we found, shown on the left in red, and one of the most significant sensitivity biomarkers we found are these custom transcriptional signatures that we derived. So again, at this phase of the project, we would then put these signatures through robustness testing to make sure we feel confident and our clients feel confident carrying the signature forward into clinical populations analysis. And just to give a generalization of an idea of a type of cutoff we would use in the robustness analysis, we would want an accuracy of at least 70% before moving forward. So again, I'm going to highlight our Uber analysis here as an example of what we would do in clinical populations analysis using these custom transcriptional signatures. And so now on the y-axis, I've put our CI1040 sensitivity signature expression. And on the x-axis, I have the CI1040 resistance signature expression. Again, each of the points on this graph represents a clinical sample that is colored by cancer type. And the lines on the graph indicate pan-cancer median expression of our CI1040 sensitivity and resistance signatures. So again, using this analysis, we're able to quickly focus in on the upper left quadrant, which are the clinical samples we would say are expressing optimal CI1040 signature expression. And again, what I mean by this is that they have high sensitivity signature expression, comparatively pan-cancer, and low resistance signature expression. So what I hope you can see on your screens is that we have some cancer types that are confined within this upper left-hand quadrant. And this includes, colored in orange is colorectal cancer, and colored in this light tan color, we have melanoma. And so we would expect, based on using a MEK inhibitor here and the types of aberrations that are common within colorectal and melanoma cancers, it's a nice proof of concept that these are falling within the upper left-hand quadrant. What perhaps you can also see is that we have some cancer types that are falling not totally within this upper left-hand quadrant. So they kind of, for example, if you look at ovarian cancer, it kind of sits right in the middle of this Uber analysis. And so if we had a hypothetical client who wanted to use a MEK inhibitor, wanted to use CI-1040 in ovarian cancer, what we're able to do within this analysis is filter down so that now we're looking at just ovarian cancer samples. And what I did was I colored these ovarian cancer samples by cancer subtype. And I tried to make sure that I colored all of the serous ovarian cancers a shade of blue. So using this analysis, if we did have a client who wanted to go into ovarian cancer, we would have the hypothesis that at least a portion of serous ovarian cancers would be sensitive to MEK inhibition. And this is because a portion of these cancers do lie within this upper left-hand quadrant. Within this analysis, we're also able to interrogate a number of different metadata within the Uber analysis. An example of this metadata would be grade of the clinical sample. And so what I'm showing in the table on the right is I queried the Uber database to ask what portion of serous endometrial cancers, what grade are those cancers that fall within this upper left-hand quadrant that has the favorable signature expression. And that's what I'm showing you in this table. So the frequency indicates the frequency at which each grade of serous ovarian cancer, falls within this upper left-hand quadrant. And then I'm providing for you the count of samples that falls in the upper left-hand quadrant, as well as the total number of samples that were interrogated. So you can see that we had 79.3% of grade 1 serous ovarian cancers fall within that upper left-hand quadrant. So that would give us the hypothesis that low-grade serous ovarian cancer is potentially sensitive to MEK inhibition. This is quite interesting in light of the fact that AstraZeneca's MEK inhibitor, selumetinib, was published as the first drug to help patients with recurrent low-grade serous ovarian cancer. Kind of a nice proof of concept of the type of hypotheses we can derive using OncoPredictor analysis and their potential usefulness in the clinic. I'd like to provide for you one more hypothetical example of, again, if we had a client that came to us that wanted to use CI-1040 now in leukemias. So again, now I've filtered down the Uber database to just show leukemias, and now I'm coloring by subtype of leukemias. And again, just as a reminder, I have the sensitivity signature on the y-axis and the resistance signature on the x-axis. So again, just looking quickly at this plot that we have here, within the upper left-hand quadrant, we have a majority of acute myeloid leukemias. So again, if our client came to us and said they wanted to use a MEK inhibitor in leukemias, what cancer subtype do you hypothesize would be sensitive to that? We would point them towards acute myeloid leukemia. Phase 2 trial results of AstraZeneca's MEK inhibitor selumetinib in advanced AML suggested that using KIT SNP as a biomarker may associate with anti-leukemic activity of selumetinib. And so I think this highlights nicely the potential usefulness of these signatures to quickly focus a drug development program down to a cancer subtype of interest, in this case AML, and then biomarker refinement could then be done within this much smaller clinical population rather than going into a general population trying to identify predictive biomarkers without any sort of stratification. And so with that, I'd like to wrap up the first portion of my talk, where I have highlighted for you our OncoPredictor analysis and our ability to generate transcriptional signatures doing this, using this analysis. And I just wanted to mention briefly that we have done a number of these proof of concept analyses to show that we can successfully identify patient populations that have been shown to respond to FDA-approved targeted agents. And I've listed for you here a number of key examples. These include the fact that we've identified HER2-positive breast cancer as being sensitive to lapatinib, BCR-ABL positive CML as being sensitive to dasatinib, non-small cell lung cancer as being sensitive to erlotinib, and advanced hormone receptor positive and HER2 negative breast cancer as a patient population that is, potential patient population that is sensitive to everolimus. And so with that, I'm going to wrap up the portion of the talk where I talk about OncoPredictor analysis. I encourage anyone that's interested in this type of analysis to contact us for more information, or take a look at Thermo Fisher Scientific's website for more information. And so now I'm going to describe how we can use the Oncoming knowledge base as another resource to derive these transcriptional signatures. As I said before, the Oncoming knowledge base is essentially the compendium of the world's genomic data. We have been curating here for over 8 years. Not only curating, but ontologizing all of these data sets to enable interrogation across the entire knowledge base. So this knowledge base has been set up to support a number of different workflows. Today I'm going to highlight one workflow, and I hope you stay tuned for the other speakers within this series, because I know Dr. Kazanoff as well as Dr. Eddy are both going to highlight other types of workflows that can be utilized within the Oncoming knowledge base. So today I'm going to highlight the ability to use the Oncoming knowledge base to derive signatures from either single or multiple data sets located within Oncoming. And today I'm actually going to give the example of deriving signatures from multiple data sets. So what I did for you for this talk was I derived crizotinib sensitivity and resistance signatures. And the reason I decided to focus in on crizotinib is because my colleague, Sean Eddy, is going to give a presentation today that describes an analysis where we tried to identify potential expanded clinical opportunities for crizotinib. And so I thought if I started off with a little bit of discussion of crizotinib, it would add some continuity to this seminar series. So essentially what I did was I took 3 data sets present within Oncoming that had information regarding crizotinib sensitivity. 2 of these data sets were cell line based, and 1 of the data sets was clinically based, clinical samples. And so I essentially derived sensitivity and resistance signatures within each of the data sets, and these were derived much the same way as they're derived in the OncoPredictor analysis, with essentially doing a differential expression analysis between samples designated as sensitive and samples designated as resistant. I then identified the gene overlap between the sensitivity or resistance signatures among the 3 data sets. And then I used Fisher's exact test to rank the statistical association of the genes lists. And then I used genes with a median ranking of less than or equal to 500 as the final crizotinib sensitivity and resistance signatures. And then what I could do was I could take those crizotinib sensitivity and resistance gene signatures that I derived using just the Oncoming knowledge base and upload those into the same Uber analysis that I highlighted with the OncoPredictor Analysis. And so essentially, if you look at the figure on the right, I put the crizotinib sensitivity signature expression on the y-axis and the resistance signature expression on the x-axis. And just to briefly orient you again to the Uber database, Uber is essentially a subset of the Oncoming knowledge base of samples that have been measured on U133 chips. And so on the analysis that I'm showing you here, I filtered down to just the clinical samples, and I filtered out the preclinical samples. So again, each of these points represents an individual clinical sample that is colored by cancer type. So again, that upper left quadrant, which I have indicated with a red box, is where we would say we have optimal signature expression, meaning high sensitivity and low resistance. The table on the left then provides for you the frequency at which each of these cancer types falls within that upper left-hand quadrant. And so I provide for you again the frequency in one column, the count, which are the number of samples that actually fall within the upper left-hand quadrant, and the total number of samples that have been measured. And so with this analysis, we would hypothesize that at least a portion of the samples that fall within the upper left-hand quadrant would actually be sensitive to crizotinib. And so now if I filter down to look at the proportion of cancer subtypes that fall within that upper left-hand quadrant, that's what I'm showing you within this table. These are subtypes that have at least 50 total samples measured, and I filtered down to a frequency of occurring in the upper left-hand quadrant of 12.1%. And then in the final column of this table, what I'm showing are genes that are aberrant within these cancer subtypes. And I've listed a number of crizotinib target genes, including ALK, LTK, MET, ROS1, and MST1R. And what I hope you can appreciate is that the vast majority of these cancer subtypes that fall within this upper left-hand quadrant do have at least one crizotinib target gene as being aberrant, and in most cases it's multiple of crizotinib target genes are aberrant. And what I mean by aberrant is either they have overexpression of the target gene, they have a mutation, or they have some sort of copy number aberration. And so these results kind of support for us the hypothesis that cancer types and cancer subtypes that do have this optimal signature expression and fall within the upper left-hand quadrant of our Uber analysis, at least a portion of those actually would be sensitive to crizotinib inhibition. And so again, I wanted to go back to the Uber analysis to now highlight one of these cancer subtypes that we identified in this analysis. And so I filtered down now to show you just lung cancer samples. And again, here I'm coloring now by lung cancer subtype. And what you can see is that the cancer subtype that falls within this upper left-hand quadrant, the vast majority of these samples are lung adenocarcinoma samples, with a small portion of not otherwise specified samples falling within this upper left-hand quadrant. So this would lead us to the hypothesis that at least a portion of lung adenocarcinoma samples would be sensitive to crizotinib treatment. Again, the beauty of these signatures is to really zone in quickly on cancer types or cancer subtypes that could potentially be sensitive. And then you can look deeper as to whether these cancer subtypes have additional univariate biomarkers that would confer sensitivity to the drug. And so I wanted to give an example of that within this lung adenocarcinoma population. And so what I did was I now filtered the Uber data set to show samples that have been measured for EML4 ALK gene fusions. So I've colored in pink samples in which the EML4 ALK gene fusion was found, and in blue I've colored samples where the gene fusion has not been found. And what I hope you can see is that I've blown up this upper left-hand quadrant, and you can see that these signatures do a fairly good job of identifying the vast majority of the samples that do express or do harbor the EML4 ALK gene fusion. And so I just wanted to give one final example to hopefully drive home the point that we feel these signatures really are useful in representing the biology behind what's happening after drug treatment, and are potentially very useful to identify clinical populations and subpopulations that should be sensitive to the particular drug that we're studying. So again, on the left-hand side, I'm not going to walk through it again, but this is the Uber database now. I've released all of the filters so that you can see all of the cancer types now represented. And what I'm showing on the right is that a graph that shows for you along the y-axis the frequency of favorable crizotinib signature expression. And this is essentially the frequency at which these cancer subtypes fall within this upper left-hand quadrant of the Uber database. Each of the points on the graph does represent an individual cancer subtype colored by cancer subtype. And then on the y-axis, what I'm representing here is the frequency of ALK, LTK, MET, MST1R, or ROS1. So these are all crizotinib target genes, mutation, fusion, or amplification. Excuse me, not necessarily all crizotinib target genes, all genes in which aberrations could confer sensitivity to crizotinib. And so what I'm showing by this figure is that there are three cancer subtypes that really, for us, would stand out in terms of having potential sensitivity to crizotinib. These include lung adenocarcinoma and papillary renal cell carcinoma, two cancer subtypes that are currently in clinical trials for crizotinib. And we would also pull out chronic lymphocytic leukemia as another potential cancer subtype that may show sensitivity to crizotinib. The interesting thing about this subtype is that you can see that it has very high favorable signature expression, but not necessarily mutations, fusions, or amplifications in these other potential crizotinib target genes. And so I think this helps illustrate the point that using signature expression in addition to these other univariate biomarkers is potentially helpful in shaping the form of clinical trial design. And so with that, I'd like to conclude by saying that we here at Thermo Fisher Scientific do believe in the potential for the use of predictive biomarkers to match patients to targeted therapies. This use of predictive biomarkers has the potential to revolutionize clinical trial design, the economics of healthcare, and most importantly, the future treatment of cancer patients. What I've tried to describe for you today is our belief that these transcriptional signatures do have a value in focusing in drug development programs and potentially providing a sort of differentiation in developing these drugs clinically. And what I also hope you can appreciate after listening to this talk is that the scientists here at Thermo Fisher Scientific, including myself, we do have expertise in the generation of these transcriptional biomarkers, and these can be used to support drug development programs, including helping our clients to understand a drug's mechanism of action, helping them build a competitive differentiation plan. And as I said, I didn't touch on it today, but I'd be happy to follow up with anyone who's interested. They can be used to identify potential drug combination opportunities. And so with that, I'd like to thank everyone for their time, and I'm going to see if we had any questions. It looks like we had some questions that came in while I was speaking. So the first question I see here is, within Uber analysis, are we restricted to using certain cutoffs to find cancer types with favorable signature expression? Thank you for that question, because I do realize that I highlighted throughout the talk using the pan-cancer median expression as the cutoffs each time I described the Uber analysis. We are not restricted to using the pan-cancer median cutoffs. We do typically use those as a starting point to get an idea of what cancer types and what cancer subtypes we would identify as having favorable gene expression, but we're able to customize based on our clients' needs what cutoffs we use to identify these cancer types and subtypes. So thank you, that was a good question. Let me see. We have another question here, which is, do you publish the biomarkers you identify using OncoPredictor Analysis? Thank you for that question as well. Because the majority of the work we do using OncoPredictor Analysis is proprietary client information, we don't publish and we don't typically release the data publicly, but we have had clients that we've done a number of analyses with that we do sometimes do collaborations and publish some of our results. Let me see, this question, I'm going to try to summarize. It's kind of a long question, but it says, regarding the transcriptional signatures linked to drug response in cell lines, can you elaborate on whether or not we normalize for tissue of origin when conducting the study on cell lines? Thank you for that question. That is a great question. We do take a number of steps to ensure that we're identifying biomarkers that represent the biology of the drug and not the biology of a particular cancer type. We do this in a number of different ways. For instance, we make sure that the cell line panel contains cell lines from a number of different cancer type origins. And then we have incorporated within our pipelines a number of statistical analyses that lets us quickly identify whether or not we have a cancer type or cancer subtype bias in terms of the significant biomarkers that we're pulling up. So we're able to quickly identify whether or not we potentially do have a bias, which would indicate we're identifying a biomarker that's indicative of a biology of a cancer type. And we're able to mitigate this in a number of different ways. And again, we work very collaboratively with our clients to ensure that this isn't a problem. And I think I will take one more question. Let me see. Here is a question. Have we ever been requested to evaluate more than one endpoint during the OncoPredictor analysis? Thank you for this question. Historically, yes, we have been asked to evaluate multiple different endpoints at the same time, which can be very enlightening when comparing the biomarkers that are mutually exclusive or co-occur between different endpoints. So again, I took that question because I think it highlights, again, our collaborative nature with each of the clients that we work with. And so with that, I think I would like to thank everyone again for their time. I hope you stay tuned for the next 2 talks within the series. Dr. Nick Kazanoff will follow me in a few minutes, and he'll be followed by Sean Eddy, both highlighting some of the workflows that can be used with the Oncoming knowledge base. So thank you again for your time.